In the last episode, we talked about life from the perspective of quantum physics, and after that, someone left a comment saying they never expected genetic mutation to have anything to do with quantum mechanics. To them, that idea felt almost unbelievable. But that reaction actually reveals something deeper. For a lot of people, biology, the science of life, feels completely separate from chemistry and physics. Biology seems warm, organic, alive. Chemistry and physics feel cold, abstract, mechanical, as if they belong to two different worlds. But what if they do not? What if, at the most fundamental level, biology is really just a special kind of chemistry? And if that is true, then maybe the path from non-living matter to life is not as mysterious as it seems. That is what we are going to talk about in this episode. We are going to look at a new way of thinking about life, a way in which biology and chemistry are not separate at all, but part of the same continuous process. And to understand that idea, we need to start with a field called systems chemistry. This is still a relatively new area of research, but over the past few years it has been steadily gaining ground in mainstream science. The central idea behind systems chemistry is bold, but very simple. Biology is, in essence, a branch of chemistry. From that point of view, biology is the study of highly complex chemical systems that can replicate, reproduce, and evolve. Systems chemistry, on the other hand, studies much simpler chemical systems that already show some of those same properties, especially replication. So both fields are asking similar questions, but they start from opposite ends. Systems biology usually works from the top down. It starts with living systems as we know them today, cells, organisms, ecosystems, and then tries to understand how the parts contribute to the whole. Systems chemistry goes the other way. It works from the bottom up. It starts with the simplest possible chemical systems and asks a more fundamental question. How does complexity arise in the first place? And when you apply that question to life, it becomes incredibly important. Because if life really did emerge from non-living matter, then the complexity we see in biology today was not there at the beginning. It had to be built, step by step, starting from something much simpler. That is why one of the biggest challenges in systems chemistry is figuring out the rules that allow simple chemical matter to organize itself into something vastly more complex. Most scientists agree on one basic assumption: life came from non-life. And if that is true, then life must have started from relatively simple material systems and only later developed the complexity we associate with living things today. Somewhere billions of years ago, an unknown replicating system must have appeared in a very simple form, and from that humble beginning, it followed a long, uneven path toward greater and greater complexity. Eventually, that path carried matter across a boundary that, to us, feels enormous. The boundary between chemistry and biology. And the fact that complexity seems to increase along that path suggests something important. It suggests that this was not just random drift. It suggests there may have been a driving force behind it. So, the real question is, can we explain this rise in complexity in physical terms? If life truly began from something simple, then one reasonable strategy is to study simpler systems that may preserve some of life's most basic features. Here is an analogy. Imagine you want to understand how airplanes fly. At first glance, you might think the best way is to study a Boeing 747. But that is probably one of the worst places to start. A 747 is incredibly complicated. It contains millions of parts. And many of those parts have nothing to do with the basic principle of flight. Screens, service buttons, ovens, internal systems, all of that matters for operating a modern aircraft, but none of it tells you the core reason a plane can stay in the air. If you really want to understand flight, it makes much more sense to look at an early airplane. Something simple. Something where almost every part plays a direct role. That is exactly what systems chemistry is trying to do for life. It studies simple, replicating systems, the way an engineer might study the Wright brothers' plane instead of a modern jetliner. Because if you want to understand the principle, you go back to the simplest version that still contains it. This whole bottom-up approach depends on one key assumption, that life really did begin with simple matter, and that simple matter really did pass through a process of increasing complexity. And if that is true, then bringing biology and chemistry together into one framework is not just useful, it may be necessary. Because once you place biology inside a broader chemical context, you can begin to describe life in the language of chemistry, which is often more precise and more fundamental. Now, traditionally, scientists have treated the origin of life as a two-stage story. The first stage is the chemical stage. This is the stage where non-living matter becomes simple life. The second stage is the biological stage. This is the stage where simple life becomes more complex life through Darwinian evolution. That two-part picture is very common, and at first glance, it makes sense. The first stage is usually associated with abiogenesis, the emergence of life from non-living chemistry. Somewhere in that stage, through a series of chemical transformations that we still do not fully understand, a system acquired the key property that makes life possible: the ability to replicate and evolve under its own internal dynamics. Once that happened, we could call it a simple living system. Then comes the second stage. From that point on, life grows more complex, and the story is told in the language of biological evolution. But here is the problem. What if these are not really two separate stages at all? What if this is actually one continuous process? Because if it is one continuous process, then Darwinian evolution cannot be the whole story. There must be something deeper underneath it, a more general principle, a rule that does not apply only to organisms, but also to prebiotic systems, to matter that was not alive yet, but was already moving in that direction. So, why did we ever divide the story into two parts? Honestly, the answer is pretty simple. Because we understand one part better than the other. We know much more about biological evolution than we do about the chemistry that came before it. And when one half of a process is familiar and the other half is still obscure, it is easy to draw a line between them and treat them as different worlds. But that line may say more about our ignorance than about nature itself. And ignorance is not a good basis for classification. From a chemical point of view, the process that turned non-living matter into simple life and the process that later turned simple life into complex life may be parts of the same continuous story. In fact, that is exactly what recent work in systems chemistry seems to be showing. Take a famous example. Back in the 1960s, Sol Spiegelman carried out a series of RNA replication experiments that became extremely important in origin of life research. What he showed was striking. Molecular replication could happen in a test tube. It did not require the full machinery of a living cell. That alone was a major insight. But what happened next was even more interesting. As the experiment continued, the original long RNA molecules gradually gave way to shorter RNA molecules. Why? Because the shorter ones replicated faster. And because they replicated faster, they eventually outcompeted the longer ones. So what do you see here? You see replication. You see variation. You see competition. You see selection. In other words, you see something that looks a lot like Darwinian evolution, except it is happening at the chemical level. That matters, because it suggests that natural selection is not something biology suddenly invented after life appeared. Some version of that logic may already exist in chemistry itself, and the connection goes even deeper than that. Think about ecology. Ecology feels like it should be very far away from chemistry. It deals with organisms, niches, food webs, coexistence, competition. But even there, some of the same logic shows up. Take the competitive exclusion principle. It says that if two species occupy the same niche and compete for the exact same resource, they cannot stably coexist forever. Eventually, one will outcompete the other. But if they rely on different resources, then coexistence becomes possible. Darwin's finches are one of the classic examples. On the Galapagos Islands, different finch species evolved different beaks for different food sources. Some specialized in seeds, others specialized in insects. And because they were not all competing for exactly the same thing, they could coexist. Now here is the fascinating part. Something very similar can happen with molecules. Imagine two different RNA molecules in an environment where both can replicate, but only one shared substrate is available. If one RNA species uses that substrate more efficiently, it will eventually drive the other one out. That is basically competitive exclusion, just written in chemical terms. But now change the environment. Instead of one substrate, give them 5. At first, both molecules may use those resources imperfectly and overlap in what they consume. But over time, each one may evolve toward better use of a different substrate. One becomes better at exploiting one resource. The other becomes better at exploiting another. And now suddenly, coexistence becomes possible. They no longer compete in exactly the same way, because they are no longer doing exactly the same thing. That should sound familiar, because it is the same basic logic we see in ecology, which means the behavior we associate with living populations may have much deeper roots than we thought. In a sense, Darwin's finches may simply be replaying, on a larger scale, patterns that molecules had already discovered long before birds ever existed. Now, if you step back and look at the history of life, one pattern stands out very clearly. Complexity seems to increase. The earliest life forms, around 4 billion years ago, were extremely simple. Later came eukaryotic cells, with nuclei and internal organelles. Much later came multicellular organisms, plants, animals, and everything that followed. At first glance, evolution seems to have a direction. It seems to move from the simple toward the complex. Now, that is not true for every lineage. Bacteria and archaea, for example, have remained relatively simple for billions of years. So complexity is not some universal law in the crude sense, but it is still a major feature of life's history. And if we want to understand where that trend comes from, we need to go back to chemistry. Because even though we do not know exactly what happened during the earliest stages of life's emergence, one thing is clear. The laws of chemistry were the same then as they are now. That means if we study the right kinds of chemical systems today, we may be able to glimpse the kinds of processes that mattered billions of years ago. And the right systems, in this context, are replicating molecules and the networks they form. Now here we run into a big difficulty. Getting a single molecule to replicate on its own is extremely hard, without enzymes or other biological machinery. Self-replication at the molecular level is not easy at all. But Gerald Joyce's lab found something very revealing. They worked with an RNA system in which one RNA molecule could act as a template, helping 2 RNA fragments come together and form another copy of itself. So in principle, it was self-replicating, but in practice, it was terrible at it. The replication rate was extremely slow. It took around 17 hours just to double. And worse, the process stalled after only a couple of rounds because of side reactions. So yes, self-replication was possible, but it was fragile, slow, and unstable. Then came the really interesting part. When that single molecule system was replaced by a system of 2 separate RNA molecules, the whole behavior changed. Now the 2 molecules helped catalyze each other's formation. This is called cross-catalysis. And suddenly the system became much more efficient. Instead of taking 17 hours to double, it doubled in about an hour. And as long as new reactants were supplied, replication could keep going. That is a huge clue. Because in this system, what is replicating is not really one molecule acting alone. It is the system that is replicating. And that turns out to be a very important distinction. Biology works like that all the time. A cell does not reproduce because each of its parts independently copies itself in isolation. The cell reproduces as an organized whole. So what these experiments suggest is that cooperation, or more precisely, network-level organization, can make replication dramatically more effective. And that means increasing complexity is not just some side effect. It can directly improve a system's ability to survive and reproduce. That point matters a lot, because it suggests that the transition from simple chemical replicators to biological life may have been driven, in large part, by complexification, not by complexity for its own sake, but because more complex systems can sometimes replicate more effectively than simpler ones. And once you see that, the standard sequence of evolution starts to look incomplete. Usually, we describe evolution as replication, variation, selection, evolution. But there is a missing step in that story, a crucial one: complexification. So, a better sequence might be replication, variation, complexification, selection, evolution. And that sequence may apply not just to biology, but also to the chemical stage before biology. Now, to be clear, evolution does not always move toward greater complexity. Sometimes it moves in the opposite direction. Cave animals can lose their eyesight because eyes are no longer useful in darkness. And in chemical evolution, experiments, shorter RNA molecules can outcompete longer ones simply because they replicate faster. So simplification can absolutely happen. But that does not change the deeper point. The chemical stage and the biological stage may still be governed by the same underlying logic, which brings us to a language problem. Traditionally, we describe the origin of life in chemical language at first, and then switch into biological language later. But that split can be misleading, because higher-level phenomena are often best explained in lower-level terms. Biology is usually explained through chemistry. Chemistry is usually explained through physics, not the other way around. So if the process from non-life to life is truly continuous, then maybe the whole thing should be described in chemical terms. That is where concepts like chemical kinetics become important. Chemical kinetics is the study of reaction rates, and once replication is treated as a chemical process, you can begin to analyze competition between replicators. Mathematically, you can ask how fast one system grows relative to another, how long it takes for one to dominate, under what conditions two systems can coexist. That is powerful, because chemistry is often easier to quantify than biology. And from this perspective, what biology calls natural selection may simply be the large-scale version of what chemistry already does through kinetic selection. That leads to another big question. What is the chemical equivalent of biological fitness? In biology, fitness usually means the ability to survive and reproduce. But that idea is often frustratingly vague. So, can we define something more precise? One proposal is this: the key property of replicating systems is not ordinary thermodynamic stability, but dynamic kinetic stability. That sounds technical, but the basic idea is simple. A thermodynamically stable system is one that tends to remain in a low-energy state, but a dynamically kinetically stable system is one that can maintain itself over time through continuous replication. And that kind of stability does not belong to individuals. It belongs to populations. This is extremely important. In evolution, the real persistent entity is often not the individual, but the population. Individuals come and go. The population persists through ongoing replacement. A nice analogy is a fountain. The fountain keeps its overall form, even though the individual water droplets are constantly changing. Life is like that. If you only focus on the individual, you miss something essential. This population-level way of thinking was developed in a very important form by Manfred Eigen. He studied what happens when a replicating molecule, like an RNA sequence, starts generating variants through copying errors. Some variants replicate faster, others replicate more slowly. Over time, the population explores what is called a fitness landscape. You can picture it like a mountainous terrain. The peaks represent highly successful sequences. The valleys represent poorer ones. And evolution looks like a search for higher ground. But Eigen discovered something surprising. The winner is not always a single best sequence. Often, what gets selected is a cloud of related sequences centered around a highly successful one. In other words, evolution can favor not just the fittest individual sequence, but the fittest population structure. That matters because it means diversity is not always noise. Sometimes diversity is part of what makes a system more evolvable. A mixed population can outperform a uniform one. Again, this pushes us away from thinking only in terms of individuals, and it pushes us toward thinking in terms of dynamic populations and system-level stability. Now here is the key link. If biological fitness can be reinterpreted in chemical terms. Then maybe what evolution is really doing is not maximizing fitness in some vague metaphorical sense. Maybe it is doing something more precise. Maybe replicating systems tend to move toward states of greater dynamic, kinetic stability. That gives us a candidate for the deeper driving force we were looking for. And if that is right, then natural selection is not the ultimate driver of evolution. It is the mechanism. The deeper tendency is the move toward greater dynamic kinetic stability. Natural selection helps push populations in that direction by removing forms that are worse at maintaining themselves over time. And now the whole picture starts to come together. The earliest replicators on Earth were probably incredibly fragile. Even today, chemists in advanced labs struggle to make simple molecular replicators work reliably. And yet, right outside, in an ordinary patch of soil, billions of bacteria are replicating continuously, with no scientist supervising them, no laboratory, no precision instruments, no careful experimental design, just a vast, self-maintaining population. From the standpoint of dynamic kinetic stability, that is a dramatic difference. And what bridges that difference is long-term complexification. Over immense spans of time, fragile and unstable replicators can evolve into much more stable, much more complex systems. That may be the deeper story of life. We still do not know what the very first replicator was, and we may never know. That information may be lost forever in deep time. But experiments, like Joyce's already hint at the shape of the first steps. Maybe evolution began when a replicator appeared. Maybe replication was imperfect from the start. Once replication exists, variation follows naturally. Once variation exists, interactions can produce more complex networks. Once complex networks exist, selection begins to reshape the population. And out of that repeating cycle, replication, variation, complexification, selection comes evolution. Seen this way, biology does not sit outside chemistry. It grows out of it. It is chemistry under a special condition, the condition that matter has become able to replicate. And from that point on, the story changes, because now stability no longer means just resting at lower energy. Now it can also mean maintaining a population through ongoing reproduction. That is why you can think of the material world as containing two parallel realms. There is the ordinary chemical world, and there is the world of replicators. We live in both at once. Each is governed by a different kind of stability, and the chemistry of the replicator world is what we call biology. From this perspective, we can even return to Schrödinger's famous question: how can systems far from equilibrium arise and persist? Nothing in physics or chemistry gets to violate the second law of thermodynamics. But if a replicating system acquires a way to capture energy, then something remarkable happens. It can maintain and expand its own dynamic stability while still obeying thermodynamics. In principle, even a very primitive replicator that accidentally gained the ability to harvest light or tap into some external energy source would immediately gain a major advantage over other replicators. And what is especially striking is this: that advantage might exist even if the energy harvesting replicator copied itself more slowly, because once a system can access energy, it changes the game. It gains a new way to maintain itself, a new way to stabilize its existence. And maybe that is the moment when chemistry truly begins to cross over into life. Maybe the beginning of life was not simply the first replicator. Maybe it was the first replicator that learned how to tap into energy. And if that is true, then life did not appear as a sudden miracle. It emerged as part of a continuous physical and chemical process, one driven not by mystery, but by the same kind of deep principles that shape the rest of nature.